So hello and welcome, everyone. I'd like to start our meeting by paying my respects to the First Nation custodians of country throughout New South Wales, in particular, the Gadigal people of the Eora Nation. They're the traditional owners of the land on which the University of Sydney is built. I pay my respects to their elders, past, present, and emerging. So my name is Marcella Bilek, and I'm a professor in biomedical engineering and also in the School of Physics. And I welcome you all to our biomedical engineering seminar series. We are very fortunate to have Professor Diego Mantovani joining us today. He's joining us from Canada, and his talk will be on the next generation of stents, promoting cell pro-activism through signaling. He's holder of the Canada Research Chair in Biomaterials and Bioengineering for the Innovation in Surgery from 2012 to 2026. He's a professor at the Department of Materials Engineering at Laval University, and his work aims to improve the clinical performances of medical devices for effective functional replacement and to envisage the next generations of biomaterials to enhance the quality of life of patients. He has authored more than 365 original articles, holds 5 patents, and presented more than 190 keynotes, invited, and seminar lectures worldwide in the field of advanced materials for biomedical applications. So I just ask you now to keep your mics muted and your cameras off for the duration of the talk until we reach the question and answer session. Thank you. So I'll just hand over now to Diego. Would you like to share your screen?
Actually, I was supposed that I am already sharing my screen. Can you see?
Yes, you are. I can see it, yes.
Great. I was a bit surprised for 1 second. Said, hey, everything should be in place. So thank you very much to Professor Bilek and all the department in the new department I just learned in biomedical engineering at University of Sydney. Certainly I would have a privilege to be with you, but pandemic obliging, I am still very happy to see you and to have the opportunity to share with you some of our recent advances in the field of medical devices and the next generation of stent. I am from an engineering department. I am a bioengineer by education in Polytechnic of Milan and Polytechnic of Paris in France. And I did the PhD between the University of Technology of Compiègne and Laval in medicine and polymer engineering on surface plasma modification some 22 years ago. So I am a professor since January 2000, and in general, my days are spent in a hospital where we have a laboratory. It's a research center on regenerative medicine, and we are 52 scientists between 4 faculties: dental medicine, medicine, pharmacy, and science and engineering. So first of all, thanks to my student and to a number of supporting organizations. Unhappily, I just realized that no, I never received funding from Australia. So unhappily, Australia is not yet in the list, but let's say my passion since I was a student, a PhD student, is really blood. Blood, cardiac, vascular. I love the idea of this double pump animating 5 to 20 liters of liquid inside our body, let's say flowing inside a few meters of big artery and hundreds of kilometers of very small arteries up to capillary. I always like to introduce, and probably for you, I will not stay long, it's one death on 3 in the world is due to cardiac or vascular disease. When someone say vascular, in my mind appear 3 big boxes. It's cardiac, it's peripheral, so our vessels, and it is neurological. So these are one death on 3 in the world come from this. I like to start from the very beginning, and I wanted to highlight to you how an artery, when we born, is a very beautiful and complex system. It's composed basically by an internal layer, endothelial cells. The name is intima. It's a carpet. It's a carpet. It's like a mosaic. And you have endothelial cells arranged, aligned with a blood flow. Around this intima, there is a media. The media is composed by smooth muscle cells, and these smooth muscle cells are organized cylindrically. So basically they are wrapped around the cylinders, which is composed by the endothelial cells. These provide mechanical strength, rigidity, and help the pump to pulse the blood all over the peripheral system, so the body. Around the media, there is a third layer. Adventitia is mainly composed by fibroblasts with some smooth muscle cells, and they are organized radially, like a bicycle wheel. So basically, the adventitia function is anchoring the artery inside the body. The media in the main function of the media is the mechanical properties, and the intima is like a gate, is a double. First, hemocompatibility, so blood never coagulate in contact with endothelial cells, and this beautiful arrangement of very, let's say, arranged endothelial cells is able to have a permeability, a selective permeability. So basically this means that endothelial cells can allow flow, the diffusive flow around the artery to pass by opening and closing. We will see in a moment. And with age, like every tissue in our body, the artery degenerates. The degeneration is progressive, but we can certainly imagine our arteries at a very late stage in our life, not as beautiful, as perfect, and as mosaic like the initial one. So basically, the degenerative process is a process that leads to the thickening of one of the 3 layers. Depending, the most important is when the thickening is a bit, the most actually not important, the most aggressive for the health is when you have a subintimal media, let's say accumulation of cellular debris, calcium, lipids, and these create lesion on the endothelial cells, and this can lead to the progression at a severe stage of atherosclerosis. One of my colleagues in Bulgaria, in Bulgarian country, actually did some work on a fetus. He got a fetus after an abortion at 6 months old, and he cut longitudinally the femoral artery. Believe it or not, 3 months before birth, the artery of a fetus was already affected with some degenerative disease. There is certainly an heritage, genetically heritage, that is. So what I want to make clear for you is, sometimes I heard engineers saying, oh, atherosclerosis is accumulation of lipids. No, it's much more complex than that. And I would like to catch your attention on one point: the role of endothelial cells in keeping the smooth muscle cells confined into the media. As soon as a lesion appears on the endothelium, smooth muscle cells, they are muscular, so they love to proliferate. This means that their phenotype is programmed to go out of the break lesion and basically continue to grow inside. So these smooth muscle cells, the one here in red, this is a picture, obviously, this is not a photograph, but it's a synonym of the ability of the smooth muscle cells to grow and to generate new tissue and hyperplasia. Hyperplasia is an uncontrolled proliferation of, so non-desired, not expected, and not wanted. There are basically a couple of meters of vessels in our body that are higher than 6 millimeters in diameters, and they can be replaced. For all the arteries, there are some 900 kilometers of vessels, capillaries included. For all the rest of the hundreds of kilometers, no surgical treatment if you have a blood vessel to be repaired, to be replaced below 6 millimeters in internal diameter. The media remain the big focus on everything. This is a developed, let's say, vision of, and so you can see the intima layer, the medial, and the adventitia. Note that I was not fully sincere with you. There are 1 and 2 layers. These are the elastic laminas, internal and external. This elastic lamina is something absolutely crazy from a mechanical point of view, because in one side you have the media, which is very muscular with very high rigidity, so the formation is limited, and on the other side you have a regular tissue, a bit less viscoelastic, more elastic. So basically the elastic lamina is like a bumper that is able to absorb the energy produced by the media and developing around the artery. I am giving you all these details because in 5 minutes from now, we will need them in order to progress toward the next generation on devices that might be opportune and useful to repopulate arteries and arteria. In my lab, we work on 3 projects. Basically, there are some 22 PhD students, 4 to 6 postdoc, depending from funding and industrial, and 3 research associates, 1 in chemistry, 1 in biology, and 1 in physics. I am in engineering, so 3 natural scientists, 1 engineering, some postdocs, and 22 PhD students with 3 projects. 1 project is aimed to develop bioabsorbable metals. This means the next generation, not of coating, but of materials. Here, we really believe that engineering and material science and engineering can do better for clinics and for surgery rather than adapting stainless steel, titanium, nickel titanium, chrome cobaltium alloys from industrial or military purposes and going and being required to being adapted to make devices. We defend the idea that metallic alloys should and perhaps must be designed as in function of the properties that we will require when we implant. We are developing the next generation of degradable metal basically from magnesium, iron, and zinc. The degradation is programmable and generally can vary between 6 to 12 weeks, basically 3 months, up to 3 years. And depending from the alloy that you use, you can make beautiful devices very adapted for a pediatric implant, even newborn implant, some congenital disease inside the heart, or eventually some stent for people who are relatively young and that will not appreciate to keep stenting their arteries for the next 30, 40 years. The second big block is a block working on models and in vitro models from regeneration strategies, so scaffolds made of collagen and adult cells coming from human umbilical vein. And basically the idea is to create environment where to test coatings and metals. We developed bioreactors, and it's the size of a cigarette box. And the idea is to test on 7 to 10 centimeter long artery the specific response between endothelial and smooth muscle cells to coatings. I will come back to this idea at in the late steps of my presentation today. And basically, what I will bring you today is inside the nanocoating adventure and strategy. Basically, what we plan to do is we plan to. This is. I'm sorry. I need to find a way. No. There is an animation, but I am not able because one moment, I'm very sorry. Okay. So this is how a stent is implanted. Basically, a stent is a very teeny device, is mounted on a catheter and blocked onto a balloon. The balloon is put inside through the catheter. The entrance is very often radial from the arm. And when the stent is deployed, basically, it remains in place for the rest of the life of the patient. There are 3 million of stents implanted each year in the world. A stent is weighing close to 1 gram, cost more than 1,000 Canadian dollars. Basically, this means 900, 850 Australian dollars. So the market is huge, and there are some complications. So basically, what I am bringing to you today is the very early career. The first generation of stent appeared in around the 1990s. This was developed through the angioplasty, which is just the use of the balloon in order to re-establish the narrowed section of the artery. When you implant a stent, this date back to 86 to 96. This was the first 10 years, and it took 10 years from the first trials up to a more generalized surgery in most of the hospitals worldwide. If starting from 1997, we have data, and we show that we have 1 million of bare metal stents implanted in North America, 1 million in Europe, and 1 million in the rest of the world. Unhappily, when a stent is implanted, so is in a stent which is implanted, in this case, for example, in coronary, is expected to make the artery heal. Practically, this works for 60 to 80% of the stent, but 20 to 40% of the stent will result in clinical complications. The main complication is related to the injury on the endothelium during the implantation, when the stent is expanded, this basically obliges the stent in a sort of cutting, lesioning the arterial wall. This generates some inflammation, probably in response to the penetration of the metallic strut to the delicate, let's say rigid, but delicate arterial wall, and this initiate an unfavorable interaction with leukocyte platelets, and this start the coagulation cascade. If a lesion appears, then we have overproliferation of smooth muscle cells. There are several strategies to decrease the stenosis, and one of the most used from 1997 to 2010 is basically the one of reducing the volume. What does it mean? This means one of the first stents on the market, the Cypher stent, was 140 micron in the structure thickness. Today, we have a stent in the market, let's say between 60 and 80. X-Science is one of the last stent from Abbott appeared a couple of years ago. Is a stent with a very thin coatings, and their strategies was decrease the structure in order to decrease the inflammatory response. Let's say decreasing the structure is a very interesting option, but is mainly related to mechanical engineering. So basically this means to designing the structure, and this designing the cutting of a mini tube to realize the stent in such a way that it might work. So let's remember, from 140 basically to 50% cutting. For 60 micron stent is extremely thin, then one of the main problems is the radiopacity. So how you implant this? Let's have a very quick look on the mechanical properties: stainless steel, chrome cobaltium, and nitinol. I would say this is my personal statistic, so this means I cannot cite this, but 60 to 70% of the stents worldwide are made of chrome cobaltium. Stainless steel was at this number 10 years ago. Today, stainless steel is 30 to 25% of the market, and the nitinol is very limited to some applications. Is a self-expandable materials that exploit a metallurgical phenomena that this is a shape memory. And let's look at chrome cobaltium. We have a very high tensile strength, basically the double of stainless steel, and 1.3, 1.4 times the nitinol, and we have an elastic modulus, one of the highest in metallic alloys. Reducing the structure thickness lead to lower the stenosis rate from 25 in average to 15%. But still, we have one stent on 6 that doesn't work. Also consider one point: stainless steel, nitinol, and chrome cobaltium, they contain chrome, they contain nickels, and these are considered allergenic, potentially cancerogenic, certainly cytotoxic, and eventually genotoxic materials. So basically, there is the potential of contamination of a patient. And that's why, starting from 2005, in the world, there was the development of drug-eluting stents. Drug-eluting stents are stents, thin or thick, then depending from companies and strategies, and even cardiologist interest or surgeon interest. But basically, these stents are, let's say, embedded with an ultra-thin polymer coating, and on the coating, generally, there are some molecules attached. We call these drug-eluting stents. These are stents are so embedded in a polymeric carrier and charged with anti-proliferative drugs. Anti-proliferative drugs, basically, there are few examples, I think 4 to 8, but basically too big family. limiting the smooth muscle cells activity or inhibiting the blood coagulations. Anepli even drug-eluting stent showing in-stent restenosis, which is a late process, while with bare metal stent we have early in-stent restenosis. So the first generation of stent is bare metal stent, clinical complication, early in-stent restenosis. This means 1 to 3 weeks. Anepli, drug-eluting stent, also receive some complication. And we started to know now, since 2017-2016, that reports in the literature show that drug-eluting stent, the fact of releasing anti-inflammatory or anticoagulant drugs for decades, for years, do not change how the biological environment react to the materials, and complication come. So there is a critical compromise in endothelium recovery due to non-specific drugs. When you have anti-proliferative cells, drugs, then this means that all, even endotheliums, will never appear. In other words, drug-eluting stents never coagulate, but never endothelialize. Biometal stents are known to rapidly endothelialize, but often coagulate after months to years. So, you know, the initial first generation, second generation, and so today we are traveling. There is also the potential allergic reaction to polymer coatings, that is how we can improve the performance of the design. Let's say it's 2-3 years that there is the next generation is being developed. This is really a hot topic in vascular and cardiovascular engineering, and the idea in changing our perspective on how materials and biology should reach an equilibrium. Surface modification remain a super interesting strategies because mainly you do not affect the mechanical properties of the device, but you mainly and actually only work on the bulk properties. So the idea is to change the surface properties, change, for example, wettability, and using functional group directly implanted inside the metals. So no polymer coatings, because polymer coating has the tendency to delaminate when stretched, when the materials is performed. And so the idea is to put together 3 features: pro-endothelialization, anti-inflammatory, and anti-thrombotic. Did you remember I said that bare metal stent, they are pro-endothelialization, but they often, let's say, coagulate. While drug-eluting stent, they never coagulate, but they never coagulate, but they also never endothelialize. So the strategy is grafting ultra-specific bioactive and very active molecules. Sirolimus and paclitaxel, they are not active. They are mainly, let's say, repulsive molecules. So the idea that I will enter now, the strategy of the process of the project was really, we mainly work on chrome cobaltium because it's the main percentage of coronary stent, and work on the metals, work on the coating deposition. So, and no, this is not us, sorry, this is what was done in the past. There are a number in the last 5 to 8, even 10 years, coating deposition developing to coat with a thin layer of polymers. this polymer. This is what happens when the polymer is deployed. So there is a high risk of cracks, delamination, and basically all stents are complex structures. So the idea in the project and in the research that I will show you in the next 20 minutes is really to work on metallic substrate, do not apply any coating, but engineer the oxide layer. Basically, this means answer the questions. Do we need a thick, a thin, or an absent oxide layer in order to improve the direct functionalization of the metals? The dream in this project was really to insert functional groups, amine, carboxylic, other groups directly into the metals. The functionalization was done by plasma treatment, and basically you are able to enter any kind of functional group. This is not a technology very much used. There are a lot of work in the literature on the functionalization of polymers, but we couldn't find any suitable research reporting functionalization by plasma of metallic substrate. So functional groups, these will be interesting because can be used to anchor points for biomolecular, and I should have written for drastically active biomolecular grafting. Personally, I love amine group because of the bioconjugation that results for attaching other molecules is very interesting. So the idea is on a reactive amine group produced and so implanted directly into the surface to attach bioactive molecules from, for example, the carboxylic termination. And to do that, so create a functional group on the substrate and then attach the biomolecules. This is quite a used strategy, the one to attach molecule to amine groups, because it's used for DNA, for heparin, hyaluronic, enzyme, proteins, and so on. In our mind, theoretically, we were much more comfortable in using a linking arm because the functional group is a very short group. And if I attach a very short peptide, then one of our questions was what guarantee that the peptides or the strongly active biological molecules will be available to cells, because cells are some millimeter distant from the implant. So that's why we explore two linking arms. One linking arm is PEG. PEG is widely used in biomaterials, is antifouling, highly hydrophilic, there is a COH terminal, and is composed by some 1,200 carbon chains, and molecules such as polylysine, RGD, heparin, hyaluronic have already been grasped. So it's molecules that already received a lot of consideration. Glutaric anhydride is a bit less studied because it's only 6 carbon chain, and there is synthesized naturally by the body, and there is also a COH terminal group, polylysine, RGD, fibronectin. For those of you that look at molecules here, you have their representation. One of the more thrilling part of this project was identifying if you want to promote endothelialization, you have to work in and act in all the signaling that take place during the first hour up to the first 100 hours of implantation between endothelial cells. It's a very rich environment, and there are people working on endothelial cell signaling for decades and even for lives. So we went to some colleagues that we know. And we found one of our colleagues, Caligiuri, in Bichat Hospital in Paris. Actually, in 2009, this clinician looked into PCAM. PCAM is a CD31 molecules, very specific to endothelial cells communications. So it's the way to which the gate between endothelial cells open and close is completely regulated by PCAM. So she looked for adhesin proteins, so endothelial cells specific, eventually platelets, but not smooth muscle cells. We want to inhibit platelet activation and aggregation. And so basically this cardiologist selected 8 amino acids. And these 8 amino acids, they called P8-RI. They patent with INSERM, which is the Health National Research Center of France, and basically they started the company. So in collaboration with this company, leveraging on some provincial and federal funding available in Canada, we multiplied by 5 the investment of this company. The idea was to graft short peptides, these 8 specific peptides, and create a biomimetic surface, strongly active, very much able to produce. The idea was so to take the metals, work on the oxide layer, and by plasma treatment with nitrogen and hydrogen insert amine group. When amine group were inserted by bioconjugation, attach the linking arm and eventually the 8 peptides. So basically, for stent applications, the fact of attaching directly on metals limit the effect of the deformation, of the corrosion, and eventually maximize the chemical composition of the interface between the coatings, the biological active coatings, and the metals. Plasma amination allows to determine a number of nitrogen groups and optimize those of them which are mainly amine group. And finally, the feasibility of grafting the molecule was assessed in Europe by our colleagues. There are some papers originated from this work, and this is something that I will be happy to share if you might be interested. So 7 steps: cleaning, electropolishing. You need to go down in roughness between 4 and 6 nanometers in order to control the oxide layer and to maximize the grafting. Finally, create amine groups and then attaching the linking arm, attaching the peptide, and evaluate all the performances. Basically, the peptide, as I showed you, P8-RI, and finally XPS, high resolution, and eventually TOF SIMS, immunostaining, fluorescence spectroscopy were used to assess their capacity of being grafted. So pre-cleaning in ultrasonic bath is super important because this allows to wash in order to have a very mirror surface, electropolish, no mistakes, no incoherencies in the surface, and then go to the plasma reactor. Basically, I think with Marcela, I am sure every one of you was super training in the understanding a bit what plasma is an ionized gas that will allow to create an equilibrium between what you etch from the surface to what you deposit. And at relatively low power and relatively low pressure, and for some 10 to 15 minutes maximum, with some gases used as a precursor, you can optimize, and you need to pump to keep the pressure controlled, and basically the condition for amination basically are here. with examination followed by 30-second post-treatment, because if you keep the amine group alive right after the opening the reactor for extracting the specimen, basically your amine group will be all gone, and practically we optimize the plasma in order to have a percentage of amine group around 1.5 to 2%. It's not the number of amine group, but it's the length of the linking arm of the following molecule that will dictate and control the capacity of cells and flowing molecules to become in contact with the peptide. The homogeneity is super important. This is top and down, this is the middle, and this is down and top of the stent. And if you need to through derivatization, you might be able to know exactly the percentage of amine group present in the surface. So for linking arm, this is what we did with PEG. Basically, we activate the carboxylic group, and finally we peptide the graft. Everything is reported, there are no real secret, and let's go in XPS. Basically, you start from electropolish condition, and then you add amine group, and when amine group, basically there is no nitrogen after electropolishing. Nitrogen on surface comes mainly from microwave plasma treatment. Percentage of amine group around 1.5, and finally you attach the PEG, and the PEG basically increase the percentage of carbon and decrease the percentage of nitrogen. This confirms that the PEG is attached. If then you attach the peptide, you can see here just from atomic concentration that basically, yes, it's feasible. You can have some peptide. And then we went in vitro, first to understand the phenotype of endothelial cells and to understand how the peptide can graft, and eventually also did the immunocytofluorescence in order to see the distribution of endothelial cells. The grafting optimization was a very tough but super pleasant task. Why? Because basically when you use glutaric acid, so very short linking arm, the short chain linking arm give, let's say, a very low degree of freedom to your peptide. So cells need really to become in contact. While when you use long chain linking arm like PEG, basically you give a much more higher degree of freedom to the peptides. So PEG confer higher degree of freedom, allowing the peptide to reorient according to endothelial cells. Thus, is expected to enhance the biological properties. Theoretically, I really imagine that the PEG is blocked on the surface, and basically the bioactive molecule is in my hands, and the PEG allow, like a sort of algae in the ocean, you can have a sort of mobility of your peptide. For in vitro results, we went to we checked the endothelial cells phenotype, and we see that the vascular cell adhesion molecule, the VCAM1, mediate the adhesion of platelet to the endothelium. So decrease the thrombogenic potential. Interleukin, which is an evidence of inflammation, decrease the inflammation, and basically the tissue factor pathway inhibitor, the TFPI, basically is the coagulation inhibitor, so inhibition of coagulation is enhanced, and finally the peptide grafting in vitro revealed that the surface was more anti-inflammatory and anti-thrombotic than basically the electropolish. This is exactly what we expected. So what is interesting is that there is a synergistic effect between the PEG and the 8 peptides. and the peptides composed by 8 amino acids. And then in vitro, you can see the endothelium, the electropolished, pegylated surface, and finally peptidic surface coating. As you can see here, in blue is the nucleus and in the green is the junction between endothelial cells. Basically, you have a very increased endothelial cells viability and anastomotic. Anastomotic, this means closer to the connection where the stent and the artery are very proximal. You have an increased endothelial cells viability. So this was all a coherent bunch of results that let us make comfortable some tests in the underflow. So basically circulating, we again on flat specimen, coated, electropolish and peptidic coating, we checked if the molecules was enough covalently, enough, let's say, strongly attached to the surface in order to be able to be retained and not washed by the flow. What is super interesting is that this is a disc at the bottom, the cell seeding was placed on the disc, and then we activate the flow. So as you can see, with the mimetic, the peptidic surface become much more biologically interesting. And the migration and adhesion of endothelial cells from aortic sources become stable. It's stable attached. And then we transfer all the work on commercial devices. Basically, what we did on flat specimen now needed to be translated into, this is a very thin stent. So 3 millimeter long, a 60 to 90 micron thick in thermostructures. So let's say even performing XPS on a structure with 60, 80, 90 micrometer thick become a relatively harsh job, because as you know better than me, the XPS windows of analysis is 300 by 300 micron, and the acquisition take already some time. So when you decrease these to 30 to 30, so you don't decrease by 10, you decrease by 100 times, then you need to increase the acquisition times of some days. And this is one of the difficulty to be the analysis of grafting directly on stent. We were obliged, also we involved Abbott, who provide a number of stents, and we were obliged to go in time of flight secondary mass spectroscopy imaging in order to be able to look, and you can see here in yellow, evidences of grafting, the grafting and so the attachment of the peptides directly on the ultra thin 80 micrometer thick section of the stent. So TOF-SIMS analysis confirmed the presence of the peptide by detecting the lysine fragment C4H8 and plus. We worked in negative and positive mode. You know, I think TOF-SIMS, basically it consists in a very, very low energy in order to really to collect and analyze fragment coming from positive and negative ions of what is grafting in the molecules. It's a very interesting work, and this allowed us to confirm that the peptide was really coating the stent. And then we went in animals. We compared our PEG-P8 array with two kinds of stent. The multi-link, which is a bare metal stent, and the X-Science, which is a drug-eluting stent. These two stents are bare metal and drug-eluting. The bare metal was considered in order to check the hypothesis. We know that bare metal stent multilink, especially in pig that never develop atherosclerosis, are interesting for up to 7 days, and the stent is endothelialized. So we expect that the PEG P8 coated stent will endothelialize as fast as the multilink. And we considered the X-Science, which is a polymer coating, 35 nanometer of fluorocarbon plus drug plus sirolimus. And at 28 days, this is known to never reach stenosis. So no coagulation is observed. So we assumed that if our stent will be able to be endothelialized in 7 days and do not restenose after 28 days, the superiority of the stent was confirmed. And
could I just say that we have only 6 minutes left and we would like to leave a little bit of time for questions?
Okay, thank you very much. Okay, I will organize.
But the great results, we're loving it.
Okay, so thank you. Because we started a bit late, so I was not stressed by this, but I will arrive very, very shortly. Thank you for reminding. So basically, these are the collected specimen, and in general, the in vivo performance are mentioned here. We took the bare metal stent, the drug-eluting stent, and the new PEG P8 peptide, and basically you can see the reendothelialization at 7 days. You see here some platelets, but no platelet on the pegylated materials, and the restenosis by OCT. So you can see here where the stent is implanted. Basically, you can see is permeables. So the reendothelialization was higher than drug-eluting stent and less thrombogenic than bare metal stent. And basically, this is the thickness of the capsule, and you see generally is a confluent red line. This means endothelialization is confluent. This is also quite mentioned here, bare metal stent, drug-eluting, and the plasma P8 and this is a live element. Here is the heart and the coronary, so is patent. And this is the internal elastic lamina and the external elastic lamina practically intact. So this means the implantation of a stent, basically this is after 28 days, do not bring any major effect or complications in the stent. And the collagen analysis with the media pneumointima is quite thick. The Duno is not abnormal. He is very quite regular, apart maybe here. And these are in general results, I think, quite promising. So what I hope I was able to show you is that surface modification can improve the performances without compromising the properties. The best amination efficiency was able to maintain corrosion and deformation resistance. A new platform was developed for the direct functionalization of metallic devices. And first we developed in flat, and then we transpose to real devices. It's a plasma-based strategy, plasma sterilized, so there are evidences of much more interesting than wet chemistry, and when coupled with CD31 peptide coming from PCAM, improve the in vitro and the in vivo performances. When compared to what exists clinically and commercially available, we can see that there is higher reendothelialization than actually it's not a DES, but bare metal stent, and antithrombotic activity marked when compared with drug-eluting stent. This is just, let's say, switch. So in general, we have a decrease in stent restenosis rate. This is what we expect from now in vitro human. This project led to a spin-off. The spin-off is in Paris, and we have a license. We have a license to them the process in order to produce the stent. And these are 4 papers that I can send you, or you can find in the literature. Is a collaborative work between our lab in Canada, let's say veterinary medicine in France, Belgium in Namur with imaging by XPS and TOF SIMS, and the laboratory for translational science in vascular disease in Paris. These are my team. This is my team a few years ago, and Sergio is the PhD student that really advanced in collaboration with a couple of others, including Vanessa and Linda, this project. Thank you very much for your attention. I think I respected the 6 minutes. So, Flori, if you have a question, I really I will be happy to answer, and I was really happy to present you this. And sorry if I took long. I should have asked at the beginning how long I have. Thank you very much. It's okay?
Yeah, Marcella should be there.
Yes. Yeah, thank you very much, Diego, for a very interesting talk. We really enjoyed the results. So we have very little time left for questions, but yeah, feel free to unmute your microphone and ask, and you can also type in the chat box if you have any questions.
So I have one that I'd like to ask, and it's about the regulatory aspects. So how have you found the issues with regards to actually attaching biomolecules to materials? Because I think in some constituencies it's regarded as a new chemical. Or could you give us a little bit of information on how you found that aspect of translation?
In this project, we didn't check nothing with a regulatory aspect. This project was mainly devoted to a proof of concept of the superiority. Now, from my experience in other devices and with other companies and regulatory, you are completely right. The question is super pertinent because in general, less additional things you put on a stent, higher is the chance to go through regulatory quite fast. However, drug-eluting stent is something largely used in the market. So drug-eluting stent, no one is scared, and they went through FDA, European Medical Agency, Health Canada, and probably also in Australia. So we don't use, apart plasma, we didn't use nothing completely, let's say, new. I expect that the kind of results that I propose to you today may be completed with some toxicity, hemocompatibility, all the ISO 10993 stuff. This must be done in bunch. But I think these results are quite convincing that it's the superiority of the peptide reach and the fastness of endothelialization is probably promising. The list of chemicals that are involved, sure that in a regulatory effort need to be clearly mentioned and defended. This means that the claims for promoting, for receiving the FDA approval, this will require times and money and the test. But I think that the idea to go in preclinical, it's something that is exploring. I know that the company just raised 12 million euros to go the first round of preclinical with the European Medical Agency. So they were able to demonstrate. They are not yet asking the results. The results will be, I think, very important to prove or not to confirm or infirm the superiority of the device. But sure, regulatory is becoming not a limiting, but a inhibiting effect, considering for company. Basically, you need to have enough funding to do in a pentaplicate, triplicate number of works and tests in humans. Did I answer?
Yes, very well. Thank you.
Great, thanks, Diego. There is another question in the chat box, Diego, from Arnold. If you can see the question, or you need me that I read it for you?
No, it's great. So the question is how the plasma treatment with CD31 become contribute to mitigating the stenosis. First of all, Arnold, the question is super pertinent. I have to be honest. We didn't evaluate. We didn't have funding to go through the restenosis mechanism and process, where we were interested into the outcome. Is restenosis present? The fact that we didn't observe any restenosis, any coagulation in the stent made us enough happy, but this do not mean that there is no chances when you implant in human. The problem is that if we want to do this kind of study, we need to work on stent at 0, implant now, collect after 1 hour, 10 hours, 100 hours. So you need a number of animals. There is something that can be done in vitro, but then is not enough representative because blood is already coagulated in order to keep it liquid. What is the driving factors observed for the restenosis? No, basically, I'm happy I don't have any driving factors. I know we never observed, we implanted in 36 dogs, and we know in triplicate, so 3 dogs for conditions, and we know that we never observed restenosis in any of the stent. This was considered positive and promising to get the preclinical openness and capacity to go preclinical. And now we are not anymore involved in this, but yeah, I will be happy if I could obtain some funding to study the etiology with this I will be very interested in going through. Did I answer?
Thank you very much, Professor Mantovani. I think this is, to me, is one of the most exciting discovery I see from the plasma physics and engineering areas in cardiovascular disease. And that's why exactly, I want to ask that question, because the restenosis is a major problem with a lot of the stentings procedures. And I've been studying the biomechanical thrombosis. So that's why I'm really keen to see, is there another exciting areas to see, is there new biomechanics, mechanobiologies are related to this stenting? But thank you for enlightening us, and especially inspiring me today. Thank you very much for coming.
Thank you for nice words. I appreciate it.
Thank you. Is there any other questions? We are already 7 minutes over our time. If no, we can officially close the session, but feel free to stay and have a chat with Diego. I'm pretty sure you still have a bit more time, as he told me. But if no, we officially close the session, but feel free to stay if you would like to have a chat with Diego. And I thank you again, Diego, for joining us, and all you for attending our seminar today. Thank you very much.
Thank you very much. Thank you very much, and bye-bye. Have a nice day. Bye.
Bye.